I'm Francis Hassard, a reader in water microbiology at Cranfield University. As part of our Environmental Biotechnology Innovation Centre, which is a BBSRC funded engineering biology hub, my work focuses on developing innovative solutions for waste and wastewater management using engineering biology. As part of EBIC, we have several themes, and one of those is theme 3. Theme 3 is looking at how do we best scale up synthetic biology solutions for integrated water and wastewater management systems. As part of this, there are a number of challenges, and so things like the inefficiency of existing resource recovery systems, how do we manage troublesome pollution issues, so things like combined sewer overflows is a big issue at the moment. And how do we minimise the current environmental footprints of some of our systems, which are quite significant. We hope that by integrating tools derived from synthetic biology, we'll be able to develop more innovative solutions that help us to achieve what we call the circular economy. That is, where we're treating waste in a way that isn't significantly causing a detriment to the environment. So our objective is to help to try to transform some of these wastes into more valuable resources, which can then help to drive that kind of economy, which will hopefully be able to scale some of our existing systems. Ultimately, it's also about enhancing the resilience of our water and wastewater systems, and we're hoping that through engineering microbial communities to be more selective and more efficient, we'll be able to do that in a much better way. There are a number of challenges associated with this approach, and so managing the complex waste streams that we get is one of the big issues. So for example, if we're trying to selectively recover metals, if we've got lots of other competing elements in there, it can make it incredibly challenging for some of these bacteria to operate. So we're being quite flexible within theme 3 to use a variety of different tools, including natural and engineered microbial communities to do the job for us. One of the key benefits is that hopefully we'll be able to target pollutants that are really quite difficult at the moment. So things like the PFAS family, these are fluorinated alkyl substances. They're the so-called forever chemicals. They're incredibly resilient and recalcitrant to degradation at the moment, and so we think that through engineering communities we'll be able to degrade these compounds in a much better way. Also, persistent environmental pollutants like microplastics are an emerged concern which we know about, but we have very little tools at the moment to deal with this at a much larger level. So we think that by engineering microbial communities, we'll be able to deal with some of these pressing environmental challenges. Within theme 3, we're hoping to have quite significant impact in how we develop new benchmarks for sustainable engineering practices within the field of environmental biotechnology generally. So we're hoping that some of the solutions that we develop will be able to be reused to revolutionise how we do waste management generally. We think synthetic biology has a real place in this to help us tackle global challenges, and things like pollution and resource scarcity are two of the big issues that we think synthetic biology or engineering biology will be able to deal with. So we hope that through engineering biology and using some of these advanced approaches, we'll be able to deliver system-level change that will benefit both industries, policymakers, but also people and communities by contributing to a much cleaner and more sustainable future. Thank you.